Riley Alicea. I actually work on campus in the cellular reprogramming laboratory, and I'm an independent researcher as well. And I'm going to talk to you today about a couple of things. One is actually focused on more on genomics, although there isn't a lot of actual genomics in it. And then this other thing that I'm working on is actually, maybe it's related to evolvability, I don't know. But if I have time, I'll get into the second part. All right, I'm going to just get into the talk. So I do this thing called cellular reprogramming. And what is that, you ask? Well, this is something that won a recent Nobel Prize in 2012. It's where we take somatic cells in animal cells, and we put transcription factors into them. We introduce them in some fashion. And we can make all different types of cells. So we can make neurons, pluripotent cells, whatever. And it depends, and you'll see it a bit, on the types of factors that you introduce. This is my little cartoon here. So this is John Grudem. So he did some of the early work with frog embryos where they were able to reprogram cells. And this is Yamanaka, who came up with the four-factor approach. And this is a IPS colony. So this is when we make pluripotent cells. This is what results, hopefully, is this colony of transformed cells. They become these pluripotent cells from somatic cells. And it takes about 2 weeks. I'll get into that in a little bit. And as you can see here, we can make all different types of cells, muscle fibers, neurons, and pluripotent cells. Well, how do we get to, I'm going to talk about some of the modeling work that I've done and some of the lab work that we've done as well. So how do we get from that to computational biology to something maybe related to evolution? So this is our direct method of cellular reprogramming. We use a retroviral vector. You can use other methods as well. And these cells that are infected, only a proportion of them transform to this new phenotype. So we're talking about efficiencies of around 3%. So you have a bunch of cells in culture, you put the factors in, and the factors in this retrovirus infect some of the cells, and then only some of those cells go on to attain this new phenotype. And the efficiency is at about 3%. This is an example, is like a time course of how this process happens. So this is a culture of fibroblast cells, which are like cells you find in the skin, in different tissues. And over time, and this is about 12 days, you can go from these cells that basically look like fibroblasts to these colonies of pluripotent cells. And so it takes about 12 days. It depends on your protocol and everything. And so the take-home message here is that we can use, maybe 4 transcription factors. We can use a cassette like this, and we can use transgenic methods to introduce this into the cells. And we can, whatever, depending on the factors we use. And these factors are things that are normally expressed in the mature cell type. So for muscle, we'll use a combination of things that are upregulated in muscle. And for IPS, we'll use a bunch of things that are upregulated in stem cells. And so we put these in, and we obtain the phenotype, but we don't know much about the intermediate steps. Like, how do you go from, forcing the overexpression of 4 transcription factors? How do you get to other types of phenotypes? So this is just an example of what they look like. So they express the different factors in different amounts. So there's a lot of diversity in this process. This is a diagram of sort of the diversity of reprogramming regimens and outcomes that you can have. So this is something that I like to show people, to show them that this is a real phenomenon, because we can take a number of different types of somatic cells, put in different combinations of factors, and make a host of different types of somatic cells, or different types of induced cells. So for example, in 1987, we were able to take a single gene using like cDNA methods to take fibroblasts and make them into muscle fibers. They just took MyoD and they put it into the,
What's a fibroblast again? Is that just like an interstitial type cell that holds stuff together?
Yeah, basically. I mean, you find them all over the body. And so you can make a host of different cell types, from macrophages, to IPS cells, to motor neurons, dopaminergic neurons, and so forth. This is something from a recent review. This is like what we call the wheel of reprogramming. So this is another fact. This is from the journal Molecular Cell last year, where they were showing how you can convert cells. This is direct conversion of lineage. So you can go from, say, like a fibroblast to any one of these neuronal cell types. And, just depending on how you change the factors, you can make a wide diversity of cells. But we actually, in our project, and this is a project that we've been trying to get funding for, but we've gotten some funding, and we actually just published a paper on it. So this is what we call a population-based approach. And this is where we take a population of cells and we consider it as a population. And we're interested in looking at the diversity of things that go on in that population and across populations. And so here you can see that there's a general response. We know if we put a retroviral vector into this genetic background, it triggers this regulatory cascade. We don't really know exactly the details of it, but we know it happens. And then it changes somehow the collective biochemical state of the cell, and you end up with this phenotypic conversion. Now, what we're interested in is actually taking different types of cells. So in our case, we're interested in different lines of fibroblasts, perhaps from different parts of the body or perhaps from different individuals. And they may have the same genetic background and they may have different genetic backgrounds. And we can do things like measure them over days and make counts as, so this is like a time series where we're sampling at different points in the time series. And we can see that this is pseudo data, but this is basically the type of data that you get, where you get variation in terms of reprogramming. So you would take, a bunch of cell cultures, you assay them at different time points, and you look at how many cells have converted out of all cells. And you find that there's a variation in this process, that on some days there might be a bunch of cells that convert, on other days there may not be very many cells that convert. And so we're actually interested in characterizing that process. What those intervals are, what sorts of time windows, and what's maybe cells from different sources or different individuals might be very good at reprogramming. And so here you have events over time. So that's one of the things we're interested in looking at. We put the 4 factors into these cells, and we look over time, and we can see this variation in the process.
Question: Are you just measuring conversion by morphology?
Well, we usually assay it using immunocytochemistry. We can also use gene expression as well. There are different ways to assay it. You don't really just look at the phenotype. You use some sort of biochemical marker. And so, I'm going to talk about our paper a little bit in a couple slides. So this is generally what people think. This model here, this is from a paper. This is like a contemporary model of what people think is happening in this process. So they're basically, it's a model where you have 2 dimensions. One axis of this model is, you ask, okay, is reprogramming a deterministic thing or a stochastic thing? And how you address that is you say, well, if you infect a population of cells and you observe them over a certain number of days, you're going to get a certain efficiency. The question is the time between the time that you infect the cells and the time that you observe reprogramming signature, which is some biochemical indicator. Is it a uniform time for all cells, or is it variable across cells? If it's a uniform time, then it's said to be deterministic. And actually, we know from experiments that really there haven't been any observed instances of this deterministic scenario. But what's much more likely is that it's a stochastic scenario, which is where if you infect a bunch of cells and you let them go over a period of time and you get this positive signal for reprogramming, that time among the cells in that population is going to be variable. So if you look across individual populations, or even if you're able to look within that single population over time, you'll see that different cells take a different amount of time, variable amounts of time. And so we don't really know why that is, but that's one of the open questions in this area. The other axis of this model is where you either this process involves a democracy of cells or maybe a very small fraction. So that is, if I do the same experiment, I infect a bunch of cells and I transform them into a new phenotype, should I expect all of the cells to have an equal chance of converting to this phenotype, or should I only expect maybe a subpopulation? Now, I said before that the efficiency is around 3%. So that's not to say that it's automatically an elite process. What this model suggests, or what people have looked at, in some cases, have found some cell types to be more democratic. I think B cells are like that. Or other cell types to be more elitist, like fibroblasts, where you infect the cells, you get a certain efficiency. The efficiency is high if you give it enough time. That suggests that any cell in the population can reprogram at the same, So it suggests that there's something about the cells that isn't variable, that they all basically have the same machinery to make this work. Or maybe it's just a subpopulation of cells that have this ability. And again, it depends on the cell type and everything else. So there's diversity in the general mechanism. There's this, we take forced transcription factors, we put them into cells, we get a variety of large-scale outcomes. And this is, of course, due to a lot of factors. I'm not going to touch on all of them here, but I just wanted to make you aware of that when I'm describing this process to you. The first one is that there is at least some sort of inherent stochasticity. So we know that, like I said, that we've never observed this process to be a deterministic process. There's always variability in the amount of time it takes for these cells to reprogram. So that means that there's something in the biochemistry variable, whether it be genomic or epigenetic, which a lot of people think it probably is. Although when you say that word, it means almost nothing sometimes. But there are fluctuations that occur in the cells that make them, that introduce transient and random noise and things like that. And then if you're reprogramming different cell types, there are different issues. So they're different, when I reprogram, say, to a stem-like cell versus a neuronal-like cell, I'm triggering different downstream gene networks. So I might be triggering the network that makes a cell, stem cell, or turns off the identity of the previous cell type. Or I might be triggering a gene regulatory network that is important in neuron making. And these are pathways that are important in development. We're actually co-opting them here to use them to directly convert the cell to a new lineage. And so it's a stochastic complex system. And it's important to remember that reprogramming takes place at multiple scales. So it could be from the genome, it could range from the genome, it could range to something like genetic, gene-gene interactions when the genes are being expressed. It could be something like, have something to do with structural remodeling of the cell, because there's a significant amount of structural remodeling that has to go on in the cell. Or it could be just, interactions with its neighboring cells. And so all these things probably play a role, but we don't really know. The take-home message, though, is that there's a lot of diversity. And so there are two puzzling experimental results here. One is this non-uniform response. So I said before that certain cell lines perform better than others. And in our case, we observed in this paper that we just had published that some cell lines or some cell types, they possess a reprogramming efficiency that is many-fold higher than other lines tested. So if I take two lines and I reprogram them using the same factors, put them in the same conditions, even if they have the same background genotype, one might perform very well and another might not perform very well at all. The question is, why is that? And it may be due to its tissue of origin, it may be due to some other factors. We don't really know, but there is this non-uniform response. It's not like you get basically the same values. You don't get a normal distribution of variation. You get this non-uniform response. The other is that sometimes when you reprogram to two different cell types, you use the same cell line, you get a high efficiency for one type and a low efficiency for another. So if I reprogram, say, from a fibroblast to a muscle fiber, that fibroblast population would be very good at that. But if I reprogram to a neuron, some type of neuron, it might be very, the same population like you heard before. And so it's something I'm calling respecification bias, which is where something is going on that's biasing that population to perform better towards one cell type than the other. And so I have a couple of hypotheses here why that might be. There's, maybe it's context-dependent selection or some sort of population process. It could be an evolved response. It could, borrow mechanisms that are already there, put there by evolution, and using them in this process. Or it could be just an adaptable response. That some cells, when they're hit with some sort of stress, are more adaptable than others. And so that carries over into this reprogramming process. So there are a lot of things to consider. A lot of people haven't thought about it in terms of, theoretical evolution or evolutionary biology. A lot of people just do the experiments and report the results, efficiencies, and so forth. So I just got a paper accepted. It's actually available online now. So it's called Defining Phenotypic Respecification Diversity using Multiple Cell Lines and Reprogramming Regimens. And we got this into Stem Cells in Development, which is journal, developmental biology journal. And so this is a graph, actually a typical result of what I was talking about, of the non-uniform responses. So these human cell lines up here, we're taking human cell lines from what essentially is the same tissue, and we're reprogramming them into two different phenotypes. So in this case, we're reprogramming the muscle. In this case, we're reprogramming the neuron. These are from variable genetic backgrounds, but basically the same tissue. This result shows a little bit of it's basically reprogramming is uniform. So if you go across the diversity of different cell lines, you'll see that it's basically a normal distribution. There might be a little, high kurtosis to the distribution. In this case, though, in the mouse case, we had cell lines that were from the same mouse, the same genetic background, but they were from all different tissues. And we know developmentally they have different, regenerative capacities in that. In this case, we actually have this non-uniform response. So we have a number of cell lines that don't reprogram at all, some that reprogram a lot, and some that show a lot of variation in reprogramming. And so that's what I mean by this non-uniform response. And we don't really know. It's almost counterintuitive. You'd expect that if you had variable genetic, if you had genetic diversity there in different individuals, that you should see this response. It's like, well, one individual has this gene for X and it, allows, the super capacity for reprogramming. That's actually not what we see. We see this sort of almost, it's backwards of what you would expect. So I expect, I kind of encourage you to check this paper out because we have a lot more things in there that we did with this. And it's like really the first paper that's characterized two types of reprogramming at the same time, done a direct comparison. And so these non-uniform responses, and this is where this systems biology and computational biology comes in. We can actually model these with statistical models. So people haven't really done this either. They've done, characterizing the cell lines. They look at the thing. They look at the efficiencies in that, but they don't really model it with any sort of formal, using any sort of formal approach. And so I've been working on using Poisson processes to model this, and also looking at like conversion rates per unit time. So I was showing you before that if you sample your culture at different points in time, you can infer sort of a rate of reprogramming. So you have all cells in the culture, you have all the cells that are positive, that's a percentage. And then if you do it over multiple times and multiple lines, you get these graphs where you have, a conversion rate per hour, basically. You can infer that for these different cell lines. And there's variability, of course, but you can see that it's very different for different cell lines. And I would point out, too, that if you take cell lines from the same tissue, it doesn't necessarily, they don't always perform in the same way. So even within a tissue, there's either some sort of diversity in that response, even within tissues, so it can't really be, well, we don't really know what what's responsible for this. But, it's a work in progress, I guess. And then you can think of this sort of accumulation model as being like a pop, where you think of like non-uniform traffic flows and uniform traffic flows. So a non-uniform process, or a non-uniform flow of traffic suggests that there's a cell-to-cell variability, and a uniform traffic flow, or a uniform response. I'm using the traffic flow analogy as a way to like kind of get at, if you're observing the flow of traffic over like a measuring post, and you see that, the traffic doesn't occur at regular intervals. You get like, pockets of traffic and pockets of clear road, and that suggests that there's some sort of cell-to-cell variability. As opposed to this model where, there's a uniform traffic flow, there's a car every certain amount of time, that suggests cell-to-cell homogeneity in that population. And so, so there are a lot of interesting things that you can do there too as well. So now I'm going to talk a little bit about this idea of efficiency. And so we take a population approach to this. And efficiency is really, when I say efficiency, it's the number of cells that reprogram based, as a proportion of the population. And so I think a lot of the models in the literature in this area, they confound the population level effects of this and the genomic mechanisms. And one example is epigenetic regulation. A lot of people assume they make a model and they say, well, it's an epigenetic phenomena. There's certain things that happen in the epigenome that account for this diversity. But they don't think about it in terms of like the epigenome across individual cells or populations. They just have a model where it's like, there's a change that goes on that allows for different efficiency. They don't really account for the cells themselves. And so we can view this from a single cell theoretical standpoint, but we can also view this as what's going on in the cell and genome. So a lot of these single cell models don't really account for diversity. They don't have diversity built into them. And so this is actually an issue of differential cell survival. So there actually is some work on measuring lineage survivability. It's a new paper by Palmer and Feldman. Really, we're looking at survivability as a way to look at like populations. It's almost like fitness, but it's not quite like fitness. Anyways, and so if we look at, what the lesson from that paper was, maybe if we look at survivability as a variable, and we look at a model that has variation built in, maybe we can get a little bit more, we can learn a little bit more about this process. So I'm going to talk a little bit about this model that I worked on and presented it at a conference. It's not like it's kind of rough and ready. I haven't really done a lot of work on it, but just letting you know about it. It's out there. It's using connectionism to look at like what happens downstream from, basically looking at regulatory networks, but looking at them in a little bit different way than a lot of people look at them. So what I'm interested in is what happens when you take two transcription factors or multiple transcription factors and you root a connectionist network with them. So you put them at the top and then you have these connections to things that are downstream. And then what happens when you take out these connections? So you change the connectivity of the network. And you can actually have things going on in the nodes too. You can have nodes that are variable. They can have different properties. So you can model variation there. But you can also model variation in terms of the connectivity. And so that's, I don't think anyone's really done that, but that's sort of something I'm looking into. And you can actually then go from the top of this to go down to the bottom and actually have, you have your standard parameters for a connectionist model that are adapted to this. And you end up with, you can actually observe what kind of cascade happens from the top down to the bottom. And you can end up with maybe, measuring changes in a phenotype from that. And so there are a couple of parameters. One is called robustness, where we actually look at if you remove these connections, what happens to the output of the model, and what happens to these internal nodes. Another measure is criticality, which is if you cut, it's basically the scope of this displacement. So if you cut these connections and you look at the output, what is the effect of, say, cutting something up here? If you cut one connection up here, does it have a wide-ranging effect downstream in these other layers? Or is it just something that's localized to the next layer? So if I cut this connection, for example, well, this actually has a wide-ranging effect. But if I cut this connection, I maybe disable this node, but it doesn't really disable anything below in the lower levels. And so that's those are sort of things you can measure with this model. And I presented it actually at the Symposium on Emerging Topics in Control and Modeling at the Coordinated Science Lab at UIUC. Let's not be confused with the Uncoordinated Science Lab. I don't know where that is. But anyways, I don't know. I got some feedback on it. People, they were interested, but not a lot of computational biologists there. It's like a mix of people from different engineering areas. But you can actually map something like this to data. And what I'm thinking about for this is that you have this space that results from this model. So in this model, we had the R variable, which was the robustness, and the S variable, which represented the scope of the displacement. And you can actually model it to what's going on with data in cell population. So here, I have a couple of examples. So in cases where the R value is low, this is all theoretical, by the way, I'm just trying to map it to data. In cases where the R value is low and the S value is low, you get this highly efficient conversion. And so ideally, if you have a high S value or a high R value, you have basically apoptosis of the cell. So they don't convert. They basically just die. And then in the middle regime here, you get these partially converted cells. And this is an example of the type of partially converted cell we often get. This is an example of a partially converted induced pluripotent cell. And this is like where you put the virus on. They either form these tight colonies like this, or they form these colonies that are very unstable, and sometimes they transform to other things. But this is where I'm going with this. Okay, and I might talk about reprogramming bias a little bit. So the idea of bias, like I said, is where you have a fibroblast, and you might convert it to the same population to two different phenotypes independently. And it might be good at one phenotype, but not be good at the other phenotype. It might be good at both phenotypes. So we can actually measure this. We can actually look at the gene expression of the input cells. We can look at downstream gene expression. So this is where you measure the cells before and after their conversion, the phenotype. You can look at phenotypic measurements. Well, they're not really phenotypic. They're immunomeasurements and other things that allow you to look at efficiency. And then there's, of course, this input variation, which is the background variation of your cells. And so this is, we get these, rank ordered plots. So we can look at a bunch of populations. We can rank them in order of their efficiency, and then we can count them over a number of replicants. And the idea is, if we do this for both cell types, if I say convert a bunch of replicants to muscle, convert them to neuron independently, and then I look at that one cell line or population and look at its rank order with respect to the population, what I should get is some sort of frequency distribution that, it's either where the cell lines usually rank at the top or usually rank at the bottom. So in this case, this cell line is actually fairly good at making muscle. It always ranks in the top 5, when you measure this over time. But it's actually not very good at making neuron. So it's actually, I would say, poor at making neuron. So this is an example of bias for muscle. Because these cell lines, I look at the efficiency, I rank order for that same cell line, and then I build a frequency distribution. But I do it for both muscle and neuron, and then I compare the distributions. And you can see that there's no overlap. That for neurons, it's always fairly poor, and for muscle, it's always fairly good. But that's not always the case. So that was kidney tissue. And then this is from heart tissue. So this is a fibroblast from heart. And now you can see that there's a little bit of difference. Our measure we actually have for muscle, it's actually quite poor. And for neuron, it's actually the response is all over the map. And we rank them, across replicates. For some replicates, it's very good at making neuron. For other replicates, it's not good at all. And so there's this overlap between the two. And that means that there may be some bias, but it's very limited. So the lesson from these graphs is that you can actually measure in a systematic way this bias. This is the overlap calculation. And then if you just take 1 minus that, you get the reprogramming bias. And then this is the example of the reprogramming bias across different tissues. So you can see 1 is a maximum, 0 is minimum. So kidney, there's very little bias. And this is just general bias. It's for heart, or for muscle and or neuron. It doesn't matter in this graph. Testes, there's a lot of bias. Kidney, there's a lot of bias. Well, kidney lines, there's a lot of bias. This one kidney line, there's not so much. So you can see there's variation in that. And so what we think is going on in terms of this bias is that you introduce your factors, either for neuron or for muscle. And then you have these changes, they're sort of first order regulatory changes. So when you put, like I showed you in the last model, and I was using a connections model, which isn't exactly how it works in biology, but you're going to take these 4 factors and put them into the cell, and they're going to immediately affect a bunch of genes downstream. And so this is this first order gene network. But then there are going to be global effects. So these genes are then going to regulate other genes, and there's going to be a cascade which results in these new phenotypes. And somewhere in this area what the cell's doing is where this bias is happening. And so we don't know where it is. And so we could look at, we could do next-gen sequencing and really get at this problem, but we haven't been able to do that yet. And it's probably going to help us to try to understand these responses a little bit better. So that's the direction we're going, is with next-gen sequencing. I think we've got the characterization down pretty well. So this is a new mode of thinking for this field. So my proposal, and a lot of people probably don't, a lot of people in the field probably don't even think about this, because I've talked to people in the field about this. They don't think about it this way. It's that cellular reprogramming, instead of being something that has a huge black box underneath it and is hard to understand outside of simple measurements of phenotype or whatever. It's a multi-level phenomenon influenced by evolution. When I say influenced by evolution, I mean there's a lot of variation that's been put there by evolution, and that is being used, or either used directly or manipulated, co-opted by the cells during this process. So standing variation may influence reprogramming efficiency, and perhaps this idea of bias as well. And so if we take cells from different tissues and bodily location, or different ages of individual, or cells with known differences in their plasticity, we might be able to uncover this variation even further. And so our diversity at the population level approach actually results in the potential to understand this great variability within determined cell types. And so there may be actually quasi-evolutionary selective mechanisms, like in terms of epigenomic state and cellular individuality, which is where cells will just do things on their own, because either they belong to a population or they maybe in spite of their population. And these sorts of things, can be understood by casting this wider net. And so now, I know that was a little bit quick. If you have any questions, you can ask me, and I'm always available. Not 24 hours a day, but so now I'm going to talk a little bit about work. This is really something that just kind of a hobby of mine almost. So how many of you, after 2008, became really interested in economics, or more interested than you may have been before? Well, yeah, probably, yeah. But anyways, I've always kind of been interested in economics, but I actually have an undergraduate degree in anthropology, so I know something about how cultural anthropologists think about value and economics, and then how economists think about value in economics, and it's not at all the same. So sometimes, if you look at an economist, they'll say, well, economics is about the efficient allocation of resources. Value is optimized by the free market. If you just let the free market work, value will be optimized. But anthropologists actually think about it a lot differently. They think about value as being relative to your society or even the thing that you're talking about. and all culture is constructed. And so these are two things that, if you take the two approaches, they don't quite mesh up. So what I'm interested in is taking these and understanding, maybe I have a computational model I'm going to introduce, and maybe like bridging these two things. So the first thing that sort of got me interested in this too was this idea of weird resource allocations. So economics is actually about allocating resources. And the question is, why are they allocated the way they are? And so sometimes you have allocations that make sense. And sometimes, like if you used an optimization algorithm, you could match up those results with something, you ran it on, you read, like people who do agronomy or whatever, and they figure out optimal ways to plant crops in that. Sometimes there are things in the world that make no sense. So one of these are cargo cults. So this is where people invest heavily in magical objects, which are perhaps commonplace in another context. And the cargo cult comes from when when the troops were running around the South Pacific during World War II, and they had their objects that the people in the South Pacific had never seen before. So they thought they were magical objects. And so they put a lot of value in these things, even though they were very commonplace in other contexts. Other examples are where people invest heavily in entertainment, and arms races where people invest in armaments and compete for this, even though it makes really no sense to do so. But also, on top of that, there's a biological substrate for economic exchange. And so we know from non-human primate societies that there are sort of pseudo-economies there where they exchange things. And they don't like, non-human primates don't like to be cheated, neither do humans. There are some papers on this that are very classic. And also, we, like non-human primates, are social. So there's a crucial element of exchange, even if it's not formally considered a market. And of course, there is a brain network, a candidate brain network, that has been found that deals in valuation. So when you have people in experiments and they're valuing things, they're exchanging money, these areas of the brain are active. And so, I'm going to get to the model in a minute, but I'm going to kind of go through this because I need you to understand why we think of value the way we do. So, how much would you have paid for a circus, Barnum and Bailey circus, circa 1889? So the answer, what they priced it at was 50 cents in U.S. currency. But could it be free? Well, they used to have an expression, a sucker born every minute. So that is sort of maybe people thought that you were paying too much for a circus. But it's, this is part of this idea of value-added relativism. So it's hard to price something that has a different value to everyone who uses it. So if I go to a carnival or a circus, 10 people are going to get 10 different values out of it because they all thought that they all have different sort of ideas of what was valuable about that experience. And so, what if we take the example of a coconut, and this is more tangible. So the thing I was talking about before was not very tangible. It was an experience of a carnival. So you think about it, you let it wash over you. It's an experience. This is a tangible product. So you think, well, gee, a coconut must have more of a consistent value to people. But actually, we know that that's not true because we know that depending on who you are, there are different. You use different elements of the coconut. So casual consumers might only value the coconut for its milk and meat, which is this inner part. The Polynesian villager might use the entire coconut for different things. And in Gilligan's Island, of course, they used it as a radio transceiver, so it had a pretty important value there. But the point is that just one object has vastly different values depending on the population of people who are using it. So then this is an example, actually, of how cultural anthropologists think of this sort of coconut-based economy. So, and notice that there are different aspects of it that overlap. So you have things like religion and ritual appeal, and coconuts being used for pure economic exchange, and then coconut agriculture, the value of coconuts to agriculture. And you notice that these things overlap. So there are different aspects of this in the overlap that are very important to the culture. So in this case, they plant coconuts and pray for rain. This is overlap between the coconut agriculture aspect and the religion and ritual aspect. And so these overlapping areas, it's not like a Venn diagram where you can find a mutual sort of value in that. These overlapping areas actually contribute to very different changes in value. It's not, it's basically calls for a different type of model than what we would generally use. You can't use like a Venn diagram, you can't use conventional set theory. So what do? So I use these things called, I actually presented on this at A-Life last summer, and this is something that I haven't really worked out a lot. It's not like something that's, it's not like a Vita, for example, but it's a geometric model, and I'm writing code for it off and on. It's this thing called contextual geometric structure. And what it is it's essentially a space that utilizes fuzzy logic or soft classification to represent the world. So in this case, we have a surface where we have different sensory inputs: vision, audition, olfaction. And that's just one example. You can use concepts for these areas, the corner of these spaces too. And the idea is that you put things in this space, and it's not additive, so you can have different, you can have assign different values for different things, and they don't have to add up to one. And the idea is that different cultures will either share these spaces, or they'll be distinct for different cultures of these agents. And so they actually doesn't require the conventional attributes of transitivity, distributivity, and symmetry, but actually it follows the tradition of cultural blends in cognitive science literature. And so this is an example of what it actually is. So it's not necessarily good for playing chess, as I showed on the bottom. Like GoFi would really beat it handily at something like Jeopardy. But what it is good for are modeling knowledge about the world and taking in sort of the biological underpinnings of that behavior, and then modeling it as a contextual system, and then looking at these allocation processes with respect to reciprocation. And so this sets up two experiments, which I'm actually not going to go through. But I actually been, and the other part of this is that you're going to use a cryptocurrency to model the things that are exchanged between these agents. So you're using these one-way functions, which are called semantic hashes. But I bring it up because computer science people might be interested. Where you have bargaining and transactions between two agents at a time. And both agents must have the same one-way function. And this one-way function then translates into a set of values. They either share values or don't share values. And that's how you get different values across populations of these agents. I haven't quite worked out how it would work in this system. But in Bitcoin, it actually works as a way to assign value to things. Bitcoin is there's been a lot of hype around it, but it's essentially one of these things called a cryptocurrency. So if you're familiar with Second Life Linden dollars, or maybe like eBay, it's a cryptocurrency where you have a currency that isn't really based on anything in the real world. It's just, something that you use in a virtual world, or something where you use where you don't really know the value of things, but you're exchanging it like money. And then this is an example up here of cryptocurrency in terms of Linden dollars. So the idea behind a cryptocurrency is you have, you take a US dollar and you change it for this currency, and it translates into a certain amount of this currency. But beyond that, it's a self-contained market. You can buy land and sell land and buy clothing in Second Life, and they all have their own values, but it's disconnected from the US dollar value. It takes on its own properties of value. So that's why it's an attractive model for this, because you can actually, you can build your own value. You don't have to rely on the dollar being the dollar or trying to map some sort of tangible value to it. In Bitcoin, they actually use, like sort of a quasi-gambling approach where you look at the probability of event, and you either take a position, you'll either buy that event happening or not, and then you'll pay market price or not for it. And then this is how they did some of the predictions during the presidential elections, is that people would bet on a certain outcome. And then the idea was that if more people bet on this outcome, it would predict what people would actually do. Now, it's not necessarily true that that was the case. Some of these systems weren't very reputable, and some of the systems aren't very, but they actually do model human behavior in a reasonable way. So I'm not going to talk about that part. So I'm going to actually mention one last thing, and this is, so we do this thing called Carnival of Evolution. It's every month, and actually Bjorn is involved in it. He's the organizer. So I did this Carnival of Evolution 58, so we do it every month. So the 58th edition, I was the host, and I did it on my blog here. And I did this, so what you do is you volunteer if you have a blog, or even if you don't, you can start a blog. It's actually very useful and fun for academics to have a blog. And what you do is you take the posts for the month, which get submitted, and you come up with a theme. So my theme was the future of evolution. And I, interested in like why futurists are so crappy at predicting the future. But it actually, in evolution, we actually can predict, like saying phylogenetics, we actually try to predict what happened or reconstruct what happened in the past. But in a sense, we're actually predicting what's going to happen in the future. Or maybe we can actually predict what's going to happen in the future. We could have like a area of science called prospective phylogenetics to predict what's going to happen. And so the whole theme of the thing was, different things that we need to account for to predict the future of evolution. And so people have done themes like this. Last month was Doctor Who. So, but I've actually been talking to people involved with sci-fi, and we've been talking about blogging and its usefulness in science. And so people are actually quite interested in blogging, and I encourage you, if you don't have a blog, to at least think about it, because it helps you communicate with people and share your ideas. And so I'm going to stop there and ask people if they have any questions. I know that was a tour de force, but...
I have a question back on your regulatory networks. It seems like for a lot of these developmental processes, you have a lot of feedback loops typically. It looks like,
Yeah,
if I was interpreting yours right, it was mostly kind of one direction.
well, the idea is that you have, and this is a very, the idea behind this model is you have, so I'm using a sort of a connectionist approach. What I'm interested in just immediately is a first pass approximation at what happens if you put factors in, you just trigger a number of different mechanisms. So in a sense, this isn't really truly what happens with genes. It's just, what happens if you have a cascade that goes through the cell. So a very abstract thing. And I mean, you could actually have feedback loops, because these can actually be bidirectional. I haven't worked out the details of that, but, to have feedback from this back to this, so you'd have more of something. Because you would have that in a gene network. But the idea is that if you cut these connections, what's the effect downstream? And you would sort of reconstruct these connections from maybe the literature, maybe what you think is going on. If you do an experiment, you see that if you induce the expression of a gene, there's a lot of stuff that goes up. You can use that as information. People have done, obviously our knowledge of gene networks is pretty incomplete anyways. So, anything might be useful. It's their tool you can use.
On this model, where would you factor in things that are not interconnected, just have a direct effect on the network in terms of a biological model?
What do you mean?
Like a microRNA or something like that?
Oh, I haven't gotten to that yet. Well, I haven't gotten to that yet. I'm trying to keep that to a minimum. Although you can't put them in. You can put them in as separate inputs or outputs. But for this, the stuff I've been working on, I haven't really accounted for that too much. But